All right, we're going to have a quick little video here on responses of cell signaling pathways. We're going to talk a little bit about specificity, scaffolding, and how a cell signaling pathway is terminated. So the first thing I want to talk about is a response. And so one of the responses, I don't have it still open, but when you look at reception, the reception PowerPoint, it shows with the intracellular receptor, it shows the ligand binding to that receptor, and then it goes into the nucleus and binds to the DNA and causes transcription to occur. And that is the majority of responses are that. It is gene expression. It is doing something to turn on a gene. And primarily what we're making with that is we're making enzymes. And so when you make an enzyme, you make the world go round because enzymes do everything. So let's say that the response that we need is mitosis. And so what the cell might do is if it gets that signal that says, hey, it's time to divide, you're getting too big, your surface area to volume ratio is not great, you need to divide. It gets that signal, and in order to divide, it has to make a bunch of enzymes that help it move through that process of mitosis. And so the signaling pathway will end with all of those enzymes being transcribed. So that would be the response. And so that's an example of how you might see gene expression as a response. In this one, we're actually seeing a couple of pathways. So we see this first pathway, that's a GPCR. The response to this is actually to activate a second pathway. So that's the response is to bind as a ligand to a new receptor and start a new response. And so that's another possibility, is to activate a second pathway. Another possibility, these calcium ions, they're going to fuse with vesicles if this is a neuron, and I don't know that it is, but I'm going to pretend it is. These calcium ions will help that vesicle get to the cell membrane and release that neurotransmitter into the synapse. So that could be, because this is another cell signaling pathway, starting with a ligand-gated ion channel. So that could be the response, that you're releasing neurotransmitters. You can rearrange the cytoskeleton of the cell, and that causes the cell to change shape. That's another thing that, and that also can be part of mitosis. That's another thing that can be our response. So there's just all kinds of ways that cells can respond. Sometimes it's apoptosis. There's a cell signaling pathway when a cell is either infected or has a bunch of mutations or for some reason is no longer healthy. It will receive a signal sometimes from within itself. It'll send itself a signal, which we call an autocrine signal, where it will tell itself to die, and so it will start that apoptosis pathway. So that would be another example of a response that you might see in a cell signaling pathway. All right, so that's really that one. I can explain how a cell might respond. So those are some examples. Another one I wanted to show you really quickly was in this PowerPoint. This is showing all of it, reception, transduction, and response. But here the response is to convert glycogen to glucose 1-phosphate, which is a part of glycolysis. So this would be, the response would be taking glycogen out of your liver and raising your blood sugar. So that's another potential response that you could see happening. All right, anyway, the next thing is specificity. So how do we make sure that we get just the right response and it is specific to what I need? So if I get scared, I don't need my pupils to constrict. I need them to dilate. I don't need my heart to slow down. I need it to speed up and give my body more oxygen and more blood flow to get more sugar and things. I need my liver to release glycogen so my blood sugar rises, so my cells have energy for me to run or fight. I don't need to digest, because if I'm about to die, digestion is the least thing I need to do. I need to set that aside and work on other things. So how do I make sure that I slow down digestion and I make my pupils dilate and I make my heart speed up and I make my liver release glycogen and all those things? Well, the short answer is that the receptors, each receptor is going to respond a particular way to a different to its ligand. So if you have an adrenaline receptor on the liver, when adrenaline binds to that receptor, the cell signaling pathway that's activated is going to end in the response of glycogen being released. If you have an adrenaline receptor on a heart, the cell signaling pathway is going to be, when that adrenaline binds to that receptor, to increase the speed of contraction. If I have a adrenaline receptor on my intestines, when that adrenaline binds, it's going to slow my intestines down so that I can I can take the energy I'm using for digestion and move it somewhere else. And so a couple of different things happen that make our response specific. One is not every cell has every receptor. So there are cells in your body that don't need to respond to adrenaline or don't need to respond to testosterone or estrogen or whatever the ligand is. They don't have a need to respond, and so they don't have that receptor. And so that's one way that we make sure it's a specific response. Only those cells that need to respond will respond because only those cells have the receptor. The second way that it's different is that every cell has different, potentially different, not different, every cell type has different pathways. So like I said, adrenaline in the liver is going to release glycogen. Adrenaline that binds to a heart cell is going to increase contractions. The reason you get different responses to the same ligand is because you have different transduction pathways. So different molecules are expressed. Every cell in your body, what makes your cells different is that they have different genes expressed. So when you look at the genes that are turned on in a liver cell, and you look at the genes that are turned on in a heart cell, they're going to be different genes. Now, both cells have all 46 chromosomes. They have all of your DNA. They could express any gene they wanted to, but they don't express them all, and that's what makes your cells different. Well, because they have different genes that are expressed, they're going to have different molecules, different enzymes, different things like that. And so those pathways are going to be different, so that when that receptor is activated in a liver cell, it's going to go through a pathway that ends with glycogen release. But when that receptor is activated in a heart cell, it has different proteins, and so it's going to respond differently because it's going to have a different pathway. And so that's kind of what this slide is talking about. You get this very specific response in each cell to each ligand. One thing also, the ligands are specific, or the receptors are specific to their ligands. So if you have an adrenaline receptor, it's not going to bind estrogen or testosterone or melatonin or cortisol or whatever. It's only going to bind adrenaline. So your receptors are very specific. Each cell only has the receptors that it needs, and then each cell has the proteins and enzymes it needs to do the pathway to get the response that it needs from that receptor. So those are just some of the different ways that you can have a specific response, that you can make sure that you have the right response to your ligands. Now, sometimes that doesn't happen, and that's when we end up with different disorders and diseases. All right, the next one is I can explain the function of scaffolding. So if you think about scaffolding, when I think about scaffolding, I think about window washers. I think about Michelangelo painting the ceiling of the Sistine Chapel. I think about people who are painting ceilings and things like that. And they're up on this scaffold, and a scaffold is just something that supports you. When we talk about as a teacher, one of the things they tell us is to scaffold your learning. In other words, teach your learning in such a way that you have the background that you need to understand the next step. This is probably most easily seen in math, right? So you have to learn this before you can learn this, and then you have to learn that before you can learn the next thing. It all builds. Well, so to scaffold just means to support with the correct, whatever do you need to support. And when we talk about a scaffolding protein, that's this pink guy here. So basically you've got these different molecules that are a part of that transduction pathway. Maybe this is kinase number 1, kinase number 2, kinase number 3. And this one is going to activate number 2, and number 2 is going to activate number 3 by phosphorylating them. And so what the scaffolding protein does is says, hey, I don't want you 3 getting scattered all over the place. It'll then take us forever to get that signal down. How about I hold y'all in place? And so it literally kind of braces them in place all together, so that once this number 1 gets activated, it can immediately activate number 2 because it's right next door. And then number 2 can immediately activate number 3 because it's right next door. And so you don't have to worry about using a second messenger, or you don't have to worry about this big old kinase molecule having to move through the cytoplasm to get to kinase number 3. And so that scaffolding protein is just providing that support to hold these molecules together so that we can increase the speed and therefore the efficiency of that pathway. And then the last one is I can describe the termination of a signal. There's a couple different ways we can terminate. Number 1, we can remove the ligand. When you take the ligand out of the receptor goes back to its inactive form, which means it no longer activates the molecules after it, so they all shut off. Another way that we can do it, if you go back and look at one of these phosphorylation cascades, if I can find one. Here's one. These phosphatases, which is what this PP stands for, they will remove that phosphate. So the kinase adds a phosphate and activates it. But this phosphatase can remove that phosphate and take it back to its inactive form. So you can use phosphatases to terminate a signal because you can shut down these active proteins and make them inactive. Again, that's a benefit of a multi-step pathway. You can shut it down at any step before you even get to the response. So sometimes you can save energy by not completing a pathway when you realize, hey, I don't need that pathway. And that's all done by termination, either by removing the ligand or by inactivating some of the molecules along the way. So those are just some things about response, specificity, scaffolding, and termination with signal transduction pathways. I hope that helps, and if you have any questions, please let me know.